of Drosophila with brown-bodied, red-eyed males. When he hybridized or intercrossed in the F1 generation, he observed that two genes, what are they? Two genes did not segregate independently, and in the F2 generation or in the F2 ratio deviated very significantly from the Mendelian phenotypic ratio 9:3:1 expected when the two genes are independent, but he doesn't get that. Morgan and his group knew that genes are, the word genes are coined by Bateson, were located on the X chromosome, and saw quickly that when the two genes, when the two genes in a dihybrid cross were situated on the same chromosome, so the proportion of parental gene combination were much higher than the non-parental type. How it will be possible, I will show in the chart. Morgan carefully studied this and understood that due to the physical association or linkage of the two genes, and coined the term linkage. Okay. So this helps to understand the physical association of genes on the chromosome, which helps to understand how a recombination or recombinants are occurring during fertilization, through the non-parental gene combination. Morgan and his group also found that when the genes, when the genes were grouped on the same chromosome, some genes were very tightly linked, and some genes are loosely linked, or somewhat a little bit, while others are loosely packed, loosely linked. So here tightly linked genes shows very less recombination, which means recombination will be very less. Formation of recombinants, will be very less. But the genes which are loosely packed in that chromosomes, formation of recombinants or recombination process will be higher. Why? Because they are loosely packed, easily can enter into the next generation and shows the variations and carrying the variations, isn't it? Loosely linked genes shows higher recombination process because they are loosely linked, hence easily can move from one generation to another generation, which is there in the crossbreed. For example, he found that, finally he concluded that genes which are linked to white and yellow were tightly linked, and hence they shows 1.3% of the recombination process. So tightly linked genes which are there in the white and the yellow colored Drosophila melanogaster. I already said in the beginning. And whereas while white and miniature wing showed 30 white and miniature wings shows 37.2% of recombination. He studied Alfred Sturtevant used the frequency of recombination. He used the frequency of recombination between gene pairs. Who is that scientist? Alfred Sturtevant, who used recombination frequently between the gene pairs on the same chromosome as a measure of the distance between the genes. So, as per his knowledge, distance between the two genes on the chromosomes can be measured. Imagine it is a chromosome. This is a gene called A, this is B. Distance between the two genes. Okay. Today, genetic maps are possible by measuring the distance between the two genes. Till now, we have discussed, okay, Morgan experiment in between the different characters of Drosophila melanogaster. Parental generation cross A, cross B. You can see here yellow and white wild type of Drosophila, in the cross B, white and miniature wild type of Drosophila. You can see the genes small y, small w, small y, small w, small y, small w. These are nothing but the genes. What are they? Genes. If parental generation is there, gamete formation occurred, fertilization was there, and F1 generation is produced from the parent generation. In the F1 generation, if you carefully see, parental type about 98.7% parental type is there. Recombinant type to 1.3% is there. How much is there? 1.3%. You can see the distance between the two genes on the chromosomes, which I mentioned here. Okay. You can see clearly the distance between the two chromosomes. Okay. So the distance between the two chromosomes very less. The distance between the two genes on the chromosomes very less, which means genes are tightly linked. So the tightly linked shows less recombinance, less recombination process, which leads to formation of less recombinants, that is 1.3%. But in the F2 generation also it occurs the same. In the cross B, if you carefully see, and female and male fruit flies, Drosophila, and parent generation it is, gamete formation occurred, fertilization was happened, F1 generation was produced in the cross B. In the cross B, if you carefully see, distance between the two genes is more and they are loosely packed. Okay, genetic map, what is that? Genetic map is very much possible because of knowing the distance between the two genes, and they are loosely packed. So because of loosely packed genes, recombinants are highly produced with 37.2%. Okay. He crossed F1 generated organisms or fruit flies. Okay, he noticed F2 generation. In the F2 generation also, the recombinants are formed with a huge percentage. By this we can understand loosely packed, tightly packed genes. Loosely packed genes on the chromosomes gives more recombinants, whereas tightly packed genes on the chromosomes which will not produce much recombinants. So this is nothing but the chromosomes, whatever there, genes whatever linked on the chromosomes. Okay, tightly linked or loosely linked, whatever it is, the linking of a gene which represents linkage. What it represents? Linkage. So remaining part will continue in the next video.